Okay, so today we're going to continue our look at speculations and investigations of dinosaur physiology, and that actually continues through next Monday. So last time, just back up once. Last time I summarized some of the lines of evidence that were initially used to suggest that dinosaurs might have been warm-blooded. So this gets us up pretty much to the early 1970s or so. And today we're going to explore information that's been suggested since. Now, one important line of evidence has been looking at an aspect of bone that we really haven't taken a look at yet. So when I introduced, fossils and fossilization, I talked about how bone was more than just a structural unit of anatomy, although that would be our main use for it. So, looking at maxillae and femora and metatarsals and things like that. But it's also a living tissue, and it responds to stresses, and it changes over time. And one of the aspects of it is that its basic structure, we call it histology, can change and be modified as it grows. So histology, the structure of a tissue, is something we examine in living organisms and something that preserves in fossil organisms. So how do we look at the internal bony tissue of a fossil organism, or for that matter, a modern organism too? Well, what you do is you take the specimen, and here's, in this case I believe it's a mosasaur, so a marine reptile, or maybe a crocodilian, I can't remember, I have to look at my notes. And they embedded it in plastic resin, so that it doesn't shatter, and then slice a little thin section, at this point, sort of a thick section out of it, right here, and then polish it down on a rock polisher until it's transparent, until it's thin enough that light can penetrate it, through it. This is a common practice in geology in general, and in this particular way of doing it is something from geology that's been adopted by paleontologists and by biologists who study bone. Of course, to do histological sections in this building, in a bioscience building, it's a lot easier. Use a microtome, that is basically a tiny little guillotine to slice off little chunks of fleshy tissue. But bone is a rocky tissue, so you need this sort of technique. And then, when you look at it through a microscope, the light will penetrate through it, and you can see internal structural details that are not noticeable from just looking at the outside of the object. So there's an entire discipline of paleohistologists who have made their career by examining aspects of bony tissue, and this includes teeth, under the microscope in order to unveil some of the aspects of a living creature. Now, let me actually back up here a bit. And so here we have a hypothetical section taken out of a bone. And the bone itself isn't just the tissue bone. It isn't just hydroxylapatite, or even just hydroxylapatite with collagen fibers weaving in between it. That's the structure, that's the material we call bone. But bone is a living tissue, has blood vessels going through it. And remember, I mentioned this early on. Bone represents a store of minerals in organisms. It's where we keep calcium and phosphate and some other minerals. And sometimes the body needs to draw on that store. And what it does is there are cells that are in our bloodstream that when the body needs extra phosphorus or extra calcium or something, they leave the blood vessel, they grab some of the bone nearby and dissolve it. So they latch onto the crystal and dissolve it. And then those minerals, those ions, are now in solution. They travel through the body and hopefully get picked up and used wherever they're needed to. Now, if all that happened was the bone got dissolved, then bones would get weaker and weaker over time. In fact, if that does happen, we have a name for it. That's what osteoporosis is. It's net bone loss. But in an organism whose system is operating properly, that bony tissue gets removed, that bony crystal gets removed, but then it gets plated back later on. So there's another set of cells, and they're carrying bits of the ions that are needed to make bone. And they get up there, and they're basically like little masons. And they put new bone back in there. So this is after the bone itself has grown. So there's another set of cells, an entirely different set of cells, that created the bone out of cartilage as it was growing to begin with. This is modifying the original bone. And so what we get are these osteons, spelled like in osteology, O-S-T-E-O-N. You'll see it spelled again. Or else we're going to see this Haversian system, and that'll be spelled out larger, that characterizes places that have been reworked. So they were produced, and then dissolved, and then redeposited. In sort of longitudinal view, these would be cylinders, but in cross-section, they look like concentric rings, because basically it's the bone being built out around the blood vessels. Okay, so all organisms, all vertebrates, need to draw on their bone in order to get those materials. But animals which are Brady metabolic, or means low metabolic rate, typically cold-blooded animals, they'll need less less of these materials per unit time than a tachymetabolic animal, than an animal with a high metabolic rate. And that's because having a low metabolic rate means they don't need as much material. Having a high metabolic rate means that the chance you're going to need extra calcium someday, extra phosphorus or something, is more likely. And so when we look at the bone, especially the later bone that's put down in the life cycle of an animal, you see a lot fewer of these secondary osteons in Brady metabolic bone, whereas you can find a whole lot of it in tachymetabolic organisms. A whole lot more of this reworking. Oh, and by the way, this big arrow here is pointing to the spot that a bone cell was in. There are actually cells, the cells that are producing the bone to begin with. They sort of get trapped inside the bony material that they produce around them. They actually have little tendrils that go out so they can get replenished while they're still alive. And they're trapped in there. We can actually see the spaces in fossil bone where those bone cells used to be. So, histologists noticed early on, histologists, physiologists, that bone texture tells you something about bone growth rate or history. The ancestral type of bone that's produced after the body is growing on a more regular basis. So this is after whatever the initial bone formation. When it's growing on a regular basis, producing zones of bone, the ancestral condition is this lamellar zonal bone. It's put down, it's not reworked, so it's primary bone, it's the first bone to put down, it's slowly deposited. And if you look at the vast majority of tetrapods, that's the only sort of bone they produce. But there are some organisms that produce sort of moderate rate bone. So fibrolamellar bone, as it's called. And this has some reworking osteons present in it. And this was produced more rapidly than lamellar zonar bone, but it's still pretty strong. And it indicates that there was a rapid growth phase. And we actually can find this, for instance, in therapsids, the group to which mammals belong. We find that in archosauriforms, the group to which dinosaurs and crocodiles belong. There are some marine Mesozoic reptiles, Mesozoic marine reptiles, we'll talk about them after the break, who show this. And then there's actually full-on Haversian bone that's highly reworked with secondary osteons. And the density of these secondary osteons increases in metabolically active animals. And today we find these in mammals and birds and basically nothing else, at least at high density. It's Haversian bone. And so recognizing this, physiologists, paleontologists starting in the 1970s said, let's take a look at what dinosaur bone looks like. And so, well, first of all, we can map onto a phylogeny. This is much more recently, this is 2017, so this is not the 1970s. and see what sort of bone we find. The ones in white, they only show lamellar zonal bone. So just to show you where we are in the tree of life, these are all creatures which are closer to crocodiliforms or to birds. So birds would be theropod dinosaurs over here. But then they are to lizards. So lizards we would find like lamellar zonal bone only. Here we see a few forms that show only the old-fashioned style bone, but a lot of them that show some of this fibrolamellar bone, but only as juveniles. And then they switch over to lamellar zonal bone later in ontogeny. So those are the gray boxes. But the ones in black show the fibrolamellar bone, the faster growing bone, throughout their ontogeny. And notice, there's Dinosauria. And they're all black, as are the immediate outgroups. So Acyllosaurus is a silosaurid. Remember the sister group to dinosaurs? Probably. And pterosaurs are the flying reptiles. We see there are some other groups here that pop in that show the fibrolamellar bone only throughout ontogeny. We'll talk about some of that on the last part of this lecture. But dinosaurs show more than that, because not only do they put down as their primary bone, fibrolamellar bone, they show a lot of secondary bone. So here's a dinosaur bone. You slice it up. You look at it under a microscope, and what do you see? You see lots of haversian canals. Lots of these secondary osteons. So highly reworked. Now, one hypothesis, so that was that people said, look, we've got all these secondary osteons. Dinosaurs show bone texture that we see today only in metabolically active animals. Well, in the 1970s, some people responded to this and said, now wait a minute. We do know that secondary osteons show up in cold-blooded animals. They just come at a lower rate. So maybe what we're seeing are individuals that are simply centuries old, and they're coming in at a very low rate, and therefore over time they can get a lot of it reworked. And that was a legitimate response at the time. But what has changed? What newer information do we have that falsifies that? Yes?
No dinosaurs really lived centuries.
No dinosaurs lived centuries. So think back to last week. We talked about how we age dinosaurs with skeletochronology. It turns out big adult dinosaurs, big adult hadrosaurs, are big adults by 7 or 10. Big tyrannosaurs are big adults by about 20. Sauropods are big adults, around 20, and they might hang around for a couple decades. There are no century-old dinosaurs. So the hypothesis was a fine one at the time, but it doesn't work now. Additionally, we find a lot of haversian bone in small dinosaurs too that probably were definitely on. So that's an example of sort of this approach to lines of evidence. It's also showing that in this part of the course, you're going to have to pull bits of information from other lectures. It's like, yes, I have to deliver information lecture by lecture because I can't just dump stuff into your brain. But it's all connected. We're going to bring back phylogeny. I just used the Plato guy. We have phylogeny coming in. We're going to have adaptations from different clades. It's all going to tie together. Much more recently, people have begun to look at the rate of bone apposition, the rate that bone is actually plated out. And this is recalculated from that through math that I will say, it is beyond, I am not a physiologist, this is beyond me. But they take evidence of the growth rate of bone as tissue and go through some calculations to approximate what the resting metabolic rate is for an organism. That is, the organism at rest, not when it's big and active. And they find, first of all, among the modern animals are the ones without the little daggers in front of them. So the daggers, think of the daggers like as if they're crosses at a graveyard, so it shows that they're dead. Or you think of the daggers, they've been killed, whichever works for you. So this stuff down in here, These are extinct forms between, just to show you, here's Crocodilus. That's the Crocodilus niloticus, the Nile crocodile. These are turtles. These are lizards. These are mammals. This is a salamander. And these are birds. Specifically, that's a mallard and a chicken. So, we see that modern-day lizards and turtles have comparable bone growth rates to salamanders, and all those groups are cold-blooded animals. We also see that the living Nile crocodile has a slightly higher, but not much higher, not statistically higher, rate. It is, too, is a cold-blooded animal. And over here we see, first of all, that birds have phenomenally high growth rates. We sort of knew that. And mammals can be varied in here, but they're definitely higher. And we see that extinct dinosaurs show either essentially mammalian rates or avian rates of bone growth. As do, in fact, some of the extinct relatives of crocodilians, and we'll get to that in a later lecture. So, if this rate of bone growth does reflect metabolic rate, this also is showing dinosaurs as having warm-blooded-like conditions. And there is Dinosauria, just to highlight that again. Or this is mapping onto that cladogram using sort of a technique, a probabilistic technique to show what the ancestral conditions probably were. And we see, high rates trending towards the bird level among theropods. We see this greenish rate for other dinosaurs and for non-dinosaurian archosauriforms, as compared to the actual cold-blooded rates we see in lizards and turtles. And in fact, these dinosaurian rates are comparable in many ways to what we see in mammals today. Now, speaking of rates, here's another way of looking at it. Before, that was specifically how the bone is being plated out inside the bone when it's initially growing. This is the body as a whole. We saw this before when I talked about skeletochronology. The maximum growth rate. So during the growth curve of the organism, when they're basically in their adolescent stage and growing the fastest, it turns out that the non-avian dinosaurs, which are the boxes and the solid line, are growing at levels comparable to marsupial mammals, placental mammals, and to precocial birds. Not as high as altricial birds, but higher than modern cold-blooded reptiles. And that we can, those rates for the modern animals are, of course, modern animals. The environment's changed, so some people argue maybe that's because the environment of the past was more favorable for dinosaurian growth, for growth in reptiles. And we do see giant reptiles in the Cretaceous might grow faster than modern ones, but they don't achieve dinosaurian rates from that same environment. Another study, about 12 years ago, took a different approach. And that is, they looked at the cost of locomotion, and basically the energy required to move an organism, and the energy that's needed for the limb muscles in bipedal dinosaurs. And they found that this was consistent with high metabolic rates. Basically, in order to move these bodies around, you couldn't do it on a slow-running engine. And in fact, if those big muscles are there, which we know they're there because we have the osteological correlates of the shape of the pelvis and the tail and the femora and so forth, they've got these big muscles. In order for those muscles to be alive, basically, they need a lot of energy. And so in their study, they found that larger, so 100 kilograms or more theropods, because they're bipedal dinosaurs, had to be endotherms. They couldn't operate with an ectothermic condition. And that depending on the nature of the model, the smaller ones were consistent with tachymetabolism, although in some models they might actually be able to operate under ectothermic conditions. So you see the models there in black rather than in the white. The white models definitely put them up in the tachymetabolic range. And I would say this says ectotherm and endotherm. I would prefer it to say Brady metabolic and tachymetabolic, because what we're really talking about here is energy demand. And so remember, when I broke it down in the lecture last time, we have things like the energy source, so that's endothermy versus ectothermy. Is it from inside the body? Well, ectothermy is it from outside the body, endothermy is it inside the body. This is a more energy use sort of thing, not an energy source. Do you have a high metabolic rate or a low metabolic rate? Another set of relatively recent examinations have looked again at bones, but this is bones in a different way. Not a bone tissue type, but at the blood flow going through bones. Now, how can we possibly look at that? Well, the blood vessels in an organism have to enter the bone and exit the bone somewhere. So think about it, of course they have to. Yes, they can't stay just in the bone. They've got to be connected to the rest of the circulatory system. And the way they do so is through this thing called nutrient foramina. So a nutrient foramen, you see a nutrient foramen right there. It's a little pore where a blood vessel would enter the bone. And then it goes and it passes through the bone, and that's where it can absorb material and deposit material and so forth. And so what Kevin Seymour and colleagues have looked at is a measure of the density of nutrient foramina as a way of expressing the blood flow going through the body. And they've manifested this as a blood flow index. But you would need to, basically, the idea of the density of nutrient foramina of dinosaurs show, first of all, among living animals, where it's more easy to test. We see cold-blooded reptiles down here. We see warm-blooded mammals here. There's a little overlap. Notice there's a log scale here, so there's a bit of a spread. Dinosaurs actually wind up having this incredibly high nutrient foramina density, far higher even than in modern mammals for the most part. It doesn't necessarily mean that dinosaurs had trans-mammalian levels of physiology, but that they certainly were capable of hitting the blood flow levels needed to be an endotherm, to have the nutrients running through to help repair the bone, to help extract the minerals when needed, and so forth. Now, you might be wondering, why aren't birds shown here? In the initial study, they couldn't actually use the bird data directly because, well, they wanted to, but they thought, okay, there's an issue here. Modern birds don't use the little nutrient foramina on the sides of the bone. They typically utilize the same openings for blood vessels that they do for their air sac system. We'll talk more about the air sac system either today or on Friday. But I've talked about it before, that saurischian dinosaurs, including birds, we've got good evidence of the air sacs in the body. In the case of birds, those air sacs invade not only the vertebrae, but they enter the limb bones. So they have great big holes on their bones. Those great big holes on their bones are for the air as well as the blood vessel. So in the initial go-through, they didn't trust themselves simply to measure the bone, the holes in the bone, because they didn't know how much of that was for air and how much of it was for blood vessels. A later study added birds to that. So birds are the ones in dark blue here. And they actually matched, and this one they could actually look at the living birds themselves. They could find the specimens and trace out the blood flow rather than just relying on the bones. So it's not quite the same thing. It's not quite comparable to looking at just the bone itself. And they found they scaled very similar to mammals. So dinosaurs still wind up being anomalously high, but certainly sufficiently capable of getting those blood flow through the body, or through the bones. So we've looked at the bones in the last lecture with sort of a full, the organism itself, its anatomy. We'll talk more about the anatomy coming up. We've looked at it from structures like blood flow. We looked at it from bone histology. We can go deeper even. We can actually look at it bone and the chemical aspect of it. And there's a whole field of isotope geochemistry and isotope paleontology. So in brief, many elements have multiple, what's called isotopes, or different so-called flavors of the same element. They don't actually taste differently, as far as I know. But what they do is they have slightly different atomic masses. Well, because you can have, I mentioned unstable isotopes, radioactive isotopes early on when I talked about radiometric dating, but there are stable isotopes as well. For instance, carbon has a couple different stable isotopes, nitrogen and oxygen. And in the case of oxygen, there's the very common oxygen-16, that's the normal one, and there's the very rare oxygen-18. Oxygen-18 is a little heavier than oxygen-16. And so biological processes tend, and for that matter, environmental processes, tend to favor 16 versus 18 in certain conditions. And one of those conditions is absorbing it when you're crystallizing a substance, in this case bone. And it's dependent on temperature. That the higher temperature, the more of one isotope than the other is favored. Now, you can take the bone of a dinosaur or other organism and you can put it in a mass spectrometer and you can find out the ratio of 18 to 16, but that in and of itself isn't sufficient. You need additional information to find out what's going on. And what these guys did in this study, and you'll see other researchers in the images to come, they compared the bone isotopes in dinosaurs to the bone isotopes of creatures from the same deposits, from the same localities, living in the same environment, for whom there's pretty good evidence they were cold-blooded. Things like modern-style crocodilians, things like fish, turtles, etc. So we would expect the cold-blooded animals to have isotopes that reflected the local environment. Because they're poikilotherms, they tend to trap local environment. If the dinosaurs had poikilothermic physiologies, we would expect them to have bone isotopes that would essentially match those of the cold-blooded animals. But in fact, at different paleolatitudes, as you can see here, so what was the latitude of the site at the time it was deposited, the dinosaurs, in this case theropods, are tracking differently than the crocodilians. So this is a very early study from 21 years ago. Here's a more recent, slightly more recent study, where here's our theoretical pattern, and that is depending upon the local environment, you might expect the ectotherms to have a higher level of oxygen-18, if it's a cooler environment, than the warm bloods. Or, in the tropical regions, to have the cold-blooded animals have a lower oxygen-18 level, because their body temperature overall would be higher because the outside environment is higher. And indeed, comparing high paleolatitudes and low paleolatitudes, we see that the dinosaurs, who are in dark, have a different pattern than the crocodilians from the same localities, the same paleo environments. And not just crocodilians, also turtles, or Chelonia is turtles. So the general pattern we find is that the oxygen isotopes indicate dinosaur body temperatures were independent of the local environment, which is a general pattern we see in endotherms. In fact, specifically, it has to do with homeothermy versus poikilothermy, or poikilothermy is tracking the local ambient temperature. Homeothermy is having a stable body temperature, typically independent of what it is outside. So those were a bunch of things that looked at individual bones, maybe in the context of other bones from the same site and so forth, but was ultimately at the level of individuals. But there are other aspects to physiology that we might want to approach that look at a community level. And this was, in fact, the big contribution of Robert Bakker. So Bob Bakker, who I mentioned last time, and I mentioned back in the history of dinosaur paleontology, was a undergraduate student of John Ostrom. And Bakker's big claim to fame, in terms of this research, was looking at predator-prey ratios. He also is a sort of a good cartoonist, and artist in general, so I'm going to use a lot of his illustrations. I have on occasion done that. And the idea here is that a Brady metabolic predator eats far less per unit time than tachymetabolic ones. So if you're a cold-blooded animal, you've got a low metabolic rate, you need less food. And so either individually, so here we have a hypothetical guard Komodo dragon and a guard dog. The guard dog requires a lot more food to feed over unit time, in this case a year, for itself and its puppies, than the guard lizard and its babies. Or we could flip it around and say that the same amount of food, so that whatever the base rate of food is, can support more Brady metabolic predators than tachymetabolic ones. So, as I think I may have used this analogy on Monday, I can't remember if I did. If you were a zookeeper and you had a fixed budget for steaks, you could have 10 times as many crocodiles as you could lions for that budget. However you want to think about what do people want to see more at your zoo. So, now, a complication of this issue is the fact that nature doesn't package animals in the same sizes at all points of Earth history. So, an 8-ton Tyrannosaurus is a lot bigger than an individual tiger. Both apex predators in their respective environments. So, Bakker's solution was to calibrate his ratios based on biomass. So figure out how much tonnage, how much kilogramage, or whatever you want to call it, of each species or each ecological type was present in a particular environment. So 8,000 kilograms of Tyrannosaurus is equivalent to 8,000 kilograms of Panthera tigris and tigers, rather than worrying about individuals. And so Bakker went and did censuses, whatever the plural of censuses, censuses I guess in English, well-known paleofaunas, assemblages of organisms, and estimated the total biomass represented among the vertebrates there. So, how many tonnage was there of Camarasaurus, how much Diplodocus, how much Stegosaurus, how much Allosaurus, etc. And then calculated the fraction of the total biomass made up of predators. So it's called predator-prey ratio, but really it's the fraction of the biomass made up of predators. The idea being, I should have made that in blue. That would have been better. Okay, that's fine. I see, yes. For tachymetabolic predators, that's fine, red's good. Tachymetabolic predators, warm-blooded predators, need more fuel, and so we would expect a lower value for the predator-prey ratio. The fraction should be lower. For cold-blooded predators, radiometabolic ones, we would expect a higher predator-prey ratio. The base volume, the unit mass of food can support more of them. Everyone see how that works? All right, so let's take a look at some data. Up until very recently, like this is the first year of GL 104, I've been able to use this slide. Although I was able to use it in one other class before this. I used to teach an honors seminar on this. It used to be, I would say, no one had actually done the math among modern wild populations to really test this. That's changed. They've done it. Came out last year. And here we see predator biomass out of, now that's slightly different. They're showing just the prey, so this is closer to true predator-prey ratios. And we have, in our world, very few places where there is a dominant predator which is Brady metabolic. But one of those places are some of the islands in Indonesia in which Varanus komodoensis, the Komodo dragon, is the apex predator, the largest living lizard, and it's cold-blooded. And we see that when we compare the biomass in communities of Komodo dragon to their prey, or over here shows a histogram, compared to mammalian, more typical faunas, like lions and tigers, or all the African large predators put together, or gray wolves, we see that there is much higher, like 10 times as much mass of Komodo dragons in their communities as there are of mammalian predators in their communities. So that works out pretty well. It shows that Bakker's ideas seem to hold true. So Bakker didn't have the data. No one had done the census data from Komodo Island to really show that. He could look at modern mammalian faunas because that data was available. He actually also looked at garden data with spiders versus insects. Spiders and insects are cold-blooded, but they're at such a literally tiny scale, one has to worry about scaling effects in terms of metabolism, so that was sort of iffy. But there were fossil cases that most people considered pretty secure, where all the creatures involved were Brady metabolic. And this is back in the late Carboniferous and early Permian periods. The time when the basal synapsids, the first members of our lineage, like the fin-necked Dimetrodon, were the apex predators. And it turns out in many of the communities he examined, these fin-necked predators were making up, between 20 and 30% or so of the total biomass, consistent with the idea that they were cold-blooded animals. He also looked at cases for tachymetabolism. Now, you could look at modern populations. For example, the Serengeti today, where predators make up about 0.3 of the biomass, 0.3% rather, not 0.3%. But one has to worry about modern studies, because first of all, no modern study is untouched by the effect of human predation and human modification of the ecosystem, and we tend not to like predators and preferentially kill them, or have historically. Additionally, we know that the fossil record is a filter, and so looking at modern census data might be a bit iffy. It would be better to compare like to like. So if you look at fossil mammalian communities, we find that they run from like a fraction of a percent to a few percent. So single digits, low single digits, good test for tachymetabolism, 20 to 30% consistent with true cold-blooded predators. How about dinosaurs? Well, the data he looked at found like less than 1% to low single digits. So his data seemed to be consistent with dinosaurs having mammalian-style predator-prey ratios. Now, note, one thing this doesn't tell us is about the physiology of the herbivores in any case. That would have to be a herbivore versus productivity test, which is theoretically quite interesting, but we're only at early stages where we might be able to get that. Now, something that he found that was interesting, and I think that a lot of people have ignored somewhat, is that the early therapsid, the non-mammalian therapsid communities, and the non-dinosaurian archosaur-dominated communities, like the Triassic, show values which are neither like modern or fossil tachymetabolic organisms, or like definite Brady metabolic organisms. But they show something intermediate, sort of like lukewarm values. So higher metabolic rates than creatures were pretty much considered cold-blooded, but not as high in terms of the metabolism, or not as low in terms of the actual ratio for these forms here, which suggests that what we're seeing is the evolution of increasing of endothermy on lineages that would eventually result in mammals or dinosaurs. So let's think about what's going on here. So the equation of aerobic respiration, we've seen it before, but it's worth noting again. Just to break that down for you again, it's glucose plus oxygen yields carbon dioxide, water, and energy, or food plus inhaling yields exhaling and energy. So we eat food, rendered here as glucose, but it's food in general. We breathe in oxygen. We don't have to deal with chemistry here, so that arrow is covering up a lot of other equations. And the byproducts are our waste byproduct of carbon dioxide and our waste byproduct of water and the energy that we want to use. So, this is the way that we get most of our work done. It's worth noting that there is another equation. Our cells can also take in that same glucose without having to incorporate oxygen in this. And we get, as a byproduct, some energy, but we also produce lactic acid as a byproduct. Now, lactic acid is a waste product. If it builds up too much, it's detrimental to an organism. In fact, it hurts. That's what hurting is. The word hurting is the English word for the feeling when we build up too much lactic acid in our muscles. At least that's one of the feelings of hurt that you get. But in a pinch, it does. And anaerobic respiration provides somewhat less energy. I mean, consider less energy than aerobic ones. But it can work when you're not oxygenating actively. Even vertebrates, we and birds and lizards, we're perfectly capable of doing anaerobic respiration. It's just we don't want to do it for too long. And additionally, after you've built up this lactic acid, it takes oxygenation to break it down. You got to break that back down into other products so it's not, hurting your system. So, do you want to be an endotherm? If you want to be an endotherm, you've got to deal with a lot of things. First of all, in order to fuel that body, you're going to increase the amount of food you eat per unit time. Because you've got to fuel that body. But that in and of itself is not sufficient. You've got to increase your oxygen intake. Because food by itself is insufficient. You need to oxygenate it. But these two by themselves are not sufficient. You need to increase the speed of distribution of the glucose and oxygen to your cells. It doesn't matter if you're getting it to your body if you can't get it to your cells efficiently. So you need to get it out there. But still, that in and of itself isn't enough. Because if you did all these first buttons, but didn't deal with the last one, you're just going to build up waste products in your body, and that's not helpful. You need to be able to deal with excess carbon dioxide that you're getting into your system. You're going to need to deal with any excess water, and you're going to need to deal with excess heat. The goal is to get some heat in your body, but if you get too much heat in your body, it has bad byproducts. So if we look at an organism whose physiology we're still not certain of, we need to be able to ascertain if they have adaptations that would allow them to do all these things. So let's think about increased food processing. Ectotherms, whole-blooded animals, typically swallow food whole, or at least with little food processing. So, here is a snake swallowing a whole rodent, and as sort of a, karmic fate, a whole snake being swallowed by a frog. So, because they don't need that energy in their system as quickly, they can afford to digest things over long periods of time. And as such as carnivores, herbivorous ectotherms can take big chunks of food, but they don't have to do a lot of processing of that plant matter. They just swallow it down and let it digest there. But if you are an endotherm, if you're tachymetabolic, if you need that energy quickly, you need adaptations to increase your food processing somehow. So if you're a carnivore, you're going to need some sort of teeth to shear or shred the meat up. You're not just going to swallow food whole, typically. You want to take it apart. So in the case, for instance, of carnivorous mammals, they typically convergently have evolved specialized shearing molars. For this course, but they're called carnassials. So if you look at the molars of your cats or dogs or whatever, they have these shearing molars. They're not grinding molars anymore. And that's why actually this lion, which is a tiger, this tiger is chewing with the side of its mouth. It's actually shearing off chunks of meat. That's how cats get off big chunks of flesh. In the case of many types of carnivorous animals, and in this case including carnivorous dinosaurs, we see what's called zyphodonty. Remember the zyphodont tooth, the blade-like serrated tooth. That's good for shearing meat. In the case of herbivores, either you have to increase your ability to do oral processing of the food, that is, chewing in some fashion or other. So we see here some form of antelope. Who are you? Are you a gazelle? I don't know antelopes that well. Doing sort of rotary motion, motion that we mammals can do pretty well. Or post-oral processing of food, that is, having a gizzard or something that breaks those particles of plants down so that they're smaller and you can digest them better. So endotherms need to be able to break this down a lot. Now, we've seen, as we walked through the diversity of dinosaurs, that carnivorous dinosaurs tend to have some sophisticated adaptations, particularly some form of specialized teeth, for dealing with tearing apart prey and not just swallowing it whole like a snake might. And that herbivorous dinosaurs have at least some form of either chewing or post-oral processing of the food, and in some cases, like ceratopsids and hadrosaurids and some of the sauropods, especially derived oral processing for chewing above and beyond what they need to, or shearing or otherwise gnawing food. Additionally, in order to make sure that you've gotten enough food into your body, you need some increased locomotor capacity. So you need to cover enough territory to forage sufficient food, either for yourself or, as we see here with the ostrich, for your babies as well. So you need to have efficient ability to cover a good area to find meat or plants or fish or whatever it is. That, first of all, to have this efficient locomotor capacity, needs to have efficient respiration and circulation to keep you going. And if the limb muscles are big enough and the amount of moving you're going to have to do anyway, it's sort of not really a catch-22, it's a necessary feedback loop. You're going to need a higher metabolic rate to move your body around enough to find sufficient food to feed your body to move around enough. And we've seen that dinosaurs show a substantial level of specialization for locomotory ability, even from the early days when we have the first striding dinosaurs. Now I'm going to wrap this up by looking a little bit about respiration, the ancestral respiration in vertebrates, and then we're going to move on to new types of respiration, specialized ones, on Friday. In primitive tetrapods, they breathe by rib breathing and air gulping. So rib breathing, muscles pull on the ribs, they expand the rib cage, the flexible lungs expand, air comes in. The ribs relax, they push the air out of the lungs, air goes out. Most advanced tetrapods do some rib breathing as well. I mean, our ribs move as well, but we have something to go above and beyond. Something that we typically don't do, but primitive tetrapods do, is simply gulp air. In fact, that's the first way that animals breathe air, which is to gulp it down. Now, animals that breathe in this fashion are subjected to what's called carrier's constraint. Carrier is a respiratory physiologist who specializes in non-mammal vertebrates. And he points out that rib breathing is reduced during walking because you're doing side-to-side motion. So the same muscles you would want to use to breathe are being employed to move your body back and forth while you're moving. And consequently, also, you're compressing one side of the body when you're moving from side to side. That's sort of compressing the air out of it and shunting it into the other lung rather than allowing you to breathe efficiently. And thus, primitive tetrapods typically hold their breath while moving. That's not to say they can't do rapid movement. This lizard is moving, it's really booking. This is way slowed down. But it's doing so anaerobically. It's not fueling its lungs while it's running. It's running anaerobically. And therefore, these guys, even when they move fast, can only do so in short bursts, and it requires long recovery times. So this is the baseline from which all specialized breathing is going to evolve within the vertebrates. And we will start next time by beginning to see how various groups of vertebrates cheat. That is, how they find ways of oxygenating the body while moving, and so that they can be running aerobically. So I will see you all on Friday.